Welcome. I'm Dr. Gregg Silverman from the New York University School of Medicine, where I'm a professor of medicine and pathology and co-director of the Musculoskeletal Center of Excellence in Manhattan, New York. And I welcome you to our program today on T cells and B cell interactions in health and disease. This is Chapter 7 of the T cell program that you've been watching. So our learning objectives today is that as a result of participating in this module, participants will be able to first, describe the interdependency of T cells and B cells in the integrated immune response. You'll also be able to define and describe the linked recognition of antigen by B cells and T cells, and identify effects on T cells that result from B cell targeting in clinical practice. You'll also postulate how B cell targeting in a condition such as multiple sclerosis could work through a T cell mediated mechanism. So to begin, let's just remind ourselves of the overall organization of the immune system. And we can see at bottom the time frame in which responses are triggered within these different segments. Now, the initial barrier and defense from immune evasion from the outside are the anatomical and physiological barriers. These include the skin, the epithelial layer with their ciliary clearance of any invaders, lysozyme and tears and saliva, and if potential invaders enter through your mouth, the low stomach pH will help, amongst other factors. These happen very quickly, but if there is some evidence of triggering of an infection or a threat of infection, the innate immune system, very ancient system, is triggered through a number of different cellular and soluble molecule-related responses occurring very quickly. Neutrophils are very prominent. Soluble molecules like complement and pentatraxins, cell-associated membrane receptors like toll-like receptors are very important, and several other factors. But it's only much later, if there has been proliferation and invasion by a microbe, does the adaptive immune system work, and those are primarily B cells and T cells, and this may take up to 96 hours to see an initial response, but sometimes much faster. So is the adaptive immune system absolutely essential for the survival of multicellular species? And the answer is no. In fact, there are many, many species that develop that don't have a single lymphocyte. So here's an example, the geoduck. Now the geoduck is an invertebrate, it's a kind of a clam, it's a mollusk, and it's a very large organism. In fact, these can grow to be over a meter long. And they don't actually have a very easy life, and they don't live in a very clean environment. They sift through muck at the bottom of streams and lakes. But however, their immune system is so successful that they can live for over 100 years. So in this case, clams that don't even have a single lymphocyte can have a very long life expectancy, and because they don't have an adaptive immune system, they never develop an autoimmune disease. So let's look at the evolution of the immune system. And you can see at the very bottom are bacteria, here shown in orange. And then with the development of more complex organisms, we see what used to be called protozoans, here called protista, and there are different varieties therefrom. And then that we actually see only much later development of the three major divisions, the plants, the fungi, and animals. And all the way at the top of the upper right-hand corner, you can see that within the animal kingdom, you have some species that have finally developed an adaptive immune system. So this is really not absolutely essential, although it provides many advantages, as we'll see. So here we see in this table, we're focusing in on the animal kingdom, and we can see at top are more ancient species. These are invertebrates, and at the bottom, the appearance of vertebrates, and at the very bottom, mammals and humans. And in fact, the species at top do not have an adaptive immune system, but nonetheless can be very successful. At bottom, the earliest appearance of the adaptive immune system is somewhere around sharks, cartilaginous fish. There's some recent papers that suggest that early lymphocytes may exist in lamprey, very recently described in the past year. And what does the adaptive immune system require? Well, it requires the capacity to rearrange germline elements into these complex antigen receptors that are called B cell receptors or BCR and T cell receptors. And we can see right at the bottom that you need a recombinase activating gene. You also need MHC molecules for presentation of antigens to T cells. And you can see at right that the innate immune system has been present in all of these species. So adaptive immunity is relatively recent in appearance. So again, to reiterate, at left, The innate immune system is very rapid. It can actually begin within minutes. The receptors that they use are actually fixed in the germline, and there's a limited number of specificities, and these do not change throughout the response. At right, the adaptive immune system, the features are that it's a slow response. It can take days or weeks. However, there are many different kinds of antigen receptors that can be generated as needed by a challenge, and this creates numerous highly selective specificities, and in fact, the dominant specificities present on lymphocytes can change during a response and can improve in affinity and specificity, and all of this go together to work hand in hand, the innate and the adaptive immune system, for the destruction of pathogens. What are the key features of the adaptive immune system? Well, firstly, it builds upon the strengths of the innate immune system. So lymphocytes have many of these same innate functions themselves, membrane-associated receptors, as well as the capacity to secrete certain factors, cytokines and chemokines, that aid communication and signaling between cells. But lymphocytes themselves have very specialized features. One is their capacity to mold very specific receptors, and they're always making new receptors through these building blocks. And that, in fact, when expressed on the surface of a lymphocyte and encountering their antigen, this will potentially stimulate the lymphocyte. It's called signal 1 in the model of Cohn and Brescher, but in fact, full lymphocyte activation for proliferation and expansion requires a second signal that for co-stimulation can occur through surface molecules or cytokines, that's signal 2. So this is the two-signal model of lymphocyte activation. And this, when successful, results in clonal expansion for an antigen-focused proliferation. And the result of this is not only to combat the threat, but also there'll be the generation of antigen-specific immunologic memory. And some of these lymphocytes may be banked away to help protect for future threats throughout a lifetime. So there are B cells and T cells, and they can have complementary receptors that work together and to make the most complete, thorough, and effective immune response. But when they're circulating through the body, how do they find each other to have the most effective response? So in fact, trafficking and coordination of response is very, very important. And when an antigen-specific B cell finds the T cell with the right kind of receptor, right kind of antigen specificity to coordinate their activities, there can be a very effective immune response. And the idea is that they become the perfect couple with superbly well-matched functions for health and longevity of the host. So one of the prime features, of course, of the adaptive immune system is immune recognition here for the T cell receptor. And actually, what we're seeing here is a schematic at left for what a major histocompatibility complex looks like on the surface of the cell. Left is a schematic, right is actually what you look at from a crystallographic solution, and this is in fact the backbone of the peptides. And what you see here is really a bed of residues from beta strands, and there are guardrails at top and bottom that keep the colored peptide. The peptide is the antigen that's going to be recognized by the T cell. This is the actual T cell receptor, shown similarly in a schematic at left. And at the top, there are the variable regions that are generated through recombination. A very immense potential repertoire can be generated. Billions and billions of different types of receptors can be generated from a more limited number of germline elements. And these fold in 3 dimensions at right with beta strands into a more complex 3-dimensional structure called a beta barrel, with the variable regions at top that are involved in actual antigen recognition, constant regions at bottom that hold them in place and stabilize the complex and actually interact with the cell membrane. So in this slide, I've turned the T cell receptor upside down. And at bottom, you can see the MHC molecules that are already loaded. And you can see how they interact with each other. So at bottom, you'd have a MHC molecule on the surface of an antigen-presenting cell, and it's already loaded with the peptide. You can see it right. And along comes a T cell with its T cell receptor to be able to recognize that very specifically. And the T cell receptor actually has contact points, some of which just recognize the guardrails and the MHC molecules, and others that are highly specific for the peptide. And this is how the specific immune recognition of T cells occurs. So B cell receptors are very, very similar. They have a similar overall organization and secondary structure. And at left, we can see the very familiar structure. Here we've taken an example of an IgG molecule. And at top, you can see the variable regions that have been generated by recombination. And in 3 dimensions at right, you can see that it's the same beta barrel structure with the tips of the variable regions that are exposed for contact with the antigen. Very similar structure. The only difference for B cells is that B cells at right can actually express these receptors on the surface of a B cell, or they can secrete them. So they're soluble receptors, and both of which can have equal ability to recognize a very specific protein antigen here that might be on the surface of a pathogen threatening the host. B cell receptors are really fantastic because they really have a much greater range of structures and motifs that they can recognize. And we're seeing at the top that those blue columns are meant to be a kind of this antigen binding site of the variable region of a BCR. And at bottom is actually the results of a surface defined from a crystal and a co-crystal with the antigen. So we see an example at left of an antibody on the surface, or BCR on the surface of a cell, recognizing a very teeny tiny hapten in red. And second, in the B area, you can see that they're recognizing a linear structure, which could be a peptide, and that fits into the groove, the yellow groove on the surface of the BCR seen at bottom. And there's 2 different views there that are shown. At right, you see a larger surface that may take up a larger area on the top of the variable region. And on the furthest right, it would actually be a very large molecule that only a small surface of it could interact with the BCR. And the BCR that has been selected actually has protruding peptides. These loops will stick out in space to actually jam into a cleft to have the strongest binding and highest affinity. So in fact, when a B cell is optimally stimulated via a BCR interaction with antigen, and then there's enough second signal, or co-stimulation, what happens? And so we see this in the first panel at left. The B cell has engaged its antigen. You can see that cross-linking of an antigen receptor. And so there's a whole process that occurs. We'll go into greater length, but that's signal 1. Signal 2 is coming from engagement with a helper T cell, which is providing a membrane-associated stimulation, CD40 ligand on the T cell, interacting, in fact, with the receptor CD40 on the B cell, and that's going to provide enough stimulation. The B cell is doing its best to support the needs of this couple by presenting antigen through its antigen receptor to stimulate the T helper cell. T helper cells also producing factors like IL-4, IL-5 or IL-6 that can all stimulate the B cell. So effective signal 1 and signal 2. That clone is so happy it's going to proliferate like crazy, seen in the middle, and the outcome is going to be some production of plasma cells that are undifferentiated, making lots of secreted antibody, as well as banking away of memory B cells, because that clone was so successful you want to access it quickly for a later immune exposure and challenge. If we think about the idea that there are many different B cell clones and there's a competition. So at top we can see that of 3 potential B cell clones with 3 different antigen receptors, only one of them has the appropriate antigen receptor shown at middle, and that will actually proliferate into daughter cells that are copies of the originating B cell. The other clones at left and right, they don't have the right receptors, so they actually die or go away. They're not really stimulated, they could care less. They actually have immune ignorance of what's going on, and the result of the stimulation to the antigen receptor, again, is the production of some memory cells and lots of plasma cells over time that are antibody-producing factories. So on the other side of the coin, what is the B cell doing for the T cell? And here we can see at upper left that a B cell will take up its appropriate antigen through its antigen receptor. The bottom left, it will actually cross-link and then be internalized. That whole complex of the BCR and the antigen is internalized. It'll be delivered to an endosome that is acidic, which will chop apart everything that's in that complex. And some of the resulting peptide pieces may randomly have enough structure and affinity that you can see that they'll actually fit into a groove of the major histocompatibility complex. Then that whole endosome will be neutralized for pH, and if that MHC is appropriate and folded and filled, it'll go to the surface, and you can see in the lower right-hand corner it'll actually provide a signal for the T cell. So there'll be this formation of a relationship between a B cell and a T cell. And the important point here is that an antigen-specific B cell can be the most efficient antigen-presenting cell in the body. And this is because a high-affinity BCR enables up to 1,000-fold more efficient antigen-specific APC function compared to the simple diffusion of peptides and proteins that professional antigen-presenting cells like macrophages and DCs are dependent on for their functioning. So, when clones of B cells and T cells with paired receptors work together, very powerful immune responses can result. So in this slide, we seek to provide an example to put it into the context of an autoimmune response. At left, we can see, in fact, that if this is perhaps a lupus patient that has antibodies that can recognize different self-components from the nucleus, certain BCR on the surface of a B cell may be able to recognize one component of a nucleosome that's been released, and a nucleosome includes different binding proteins like histones wrapped with DNA. And this is when DNA is chopped up in the nucleus and may be released. In the example at the upper left-hand corner, the B cell receptor has an antibody that's specific for histone 1 or H1, takes up, processes it in its endosome, and then delivers a peptide from histone 1 to the T cell, and the T cell is very happy as a result. The right upper hand, we'll actually see an example of a B cell that has a receptor for DNA. It can take up this same complex that has DNA and histones, and as a result will process and present the components of this nucleosome after digestion in the endosome with the same MHC presentation of H1 resulting. So different BCR specificities, but for components in the same large macromolecular complex will have the same capacity to stimulate an antigen-specific T cell. And you can see other examples below where the B cells are taking up complexes that have RNA in them and are able to present other complexes as a result. The inverse story is shown at right, where in fact a B cell that has the capacity to present H1 peptides to T cells, there are actually different peptides within that H1 protein, and it can stimulate different antigen-specific T cells. So you have this reciprocal relationship, one type of lymphocyte providing the stimulation that the other needs, and a very important role for their antigen-specific receptors, BCR or TCR. But these B cells and T cells are flying through the circulation and going through all the lymphoid tissues of the body, and the frequency of an actual specific B cell or T cell is relatively low. They can only do their job most efficiently if they can find each other. So we have an image of a conductor for an orchestra, and these are many different players that are in the immune system. And for the most efficient induction of antigen-specific lymphocyte responses, mammals have developed specialized theaters or germinal centers which involve a follicular dendritic cell in the right kind of matrix and setting. And this follicular dendritic cell has very specific components and substructures. It has these processes that allow the accumulation of antigens such that there can be effective coordinated spatiotemporal encounters. So in time and space with antigen, and this allows co-stimulation and coordinated responses between B cells and T cells. So this is a picture shown of a germinal center. It's very complex. The structure, you should appreciate the processes that are in red, the cells that are in red, those are follicular dendritic cells, and their dendritic processes have these little structures at the end called ichthyosomes that capture antigen and bank them for long periods of time, years or more. And they're there as a bait for B cells and to recruit B cells that have the right specificity that will interact through their antigen receptor. And the B cells here in this germinal center have recently proliferated, and they're called centrocytes. And they're competing with each other to have the best binding for the antigen. At bottom, you can actually see what they call centroblasts, and this is what is called the dark zone of a germinal center. The dark zone because there are a lot of nuclei, lots of cells that are proliferating. They're getting help from specific helper T cells in a very complex process. But this theater is very specialized and makes for very efficient immune responses, only appeared with the mammals appearing during evolution. All right, so BCR on clonally expanded B cells, I should also add, they diversify through hypermutation. So the centrocytes and the centroblasts, as they are stimulated by antigen helper T cells, their receptors undergo little changes, little mutations. Some of these mutations in the sequence result in better binding of the antigen. Some result in lesser binding in these daughter cells. And the point is, in fact, with better binding, there can be better competition for the antigen on the follicular dendritic cells, and so it is a competition. It's almost like a big wrestling match one-on-one, who's going to get the antigen. When they get the antigen, that provides the signal for that daughter B cell to survive. And you can see at bottom the end results are that, in fact, the losers of the contest, they die. And there's a lot of wastage in the immune system. And they die by a system called programmed cell death or apoptosis. So just to think of an analogy, it reminded me of a merry-go-round because there's a lot of motion and the B cells are proliferating and they're also migrating through the body and then through the germinal centers. So it reminded me here the motion is provided by the merry-go-round and the little girl is a B cell, daughter cell, and she has an antigen receptor which is her right hand and she's searching for the antigen. If she finds the antigen, she'll win, and the antigen here is the gold ring on the merry-go-round. So this competition is really between B cells. And shown here a little bit closer up, you can get a better sense that in a germinal center reaction, B cells with BCR of varying affinities compete to interact, and the one that wins has the highest affinity for the antigen that's arrayed on the processes of a follicular dendritic cell. In the first section, we've covered the basic functions of the adaptive immune system and how coordinated responses can come from interactions of antigen-specific B cells and T cells. Shifting our focus to better understand the implications for the pathogenesis of autoimmune disease, the table I presented here gives an example or several examples of different types of autoimmune and post-infectious responses and the different types of mechanisms that are believed to be responsible. So these are simplifications, but the important effector functions responsible for disease and tissue injury at top in the section where at left side are the titles, the names of the diseases are shown in green, are examples of diseases where the major effector function and destruction of tissue relates to antibody functions. So we have autoimmune hemolytic anemia, where autoantibodies are produced that recognize red cells and cause their lysis, autoimmune thrombocytopenic purpura, their antibodies to platelets, good pastures, their antibodies to glomerular basement membrane, and so forth. So we know in these cases the autoantibodies are the major causes of the disease, but in fact these result from interactions between B cells and T cells, and they of course are associated in many cases with inflammation, so the innate immune system is involved. The middle section are examples of diseases in which there are immune complexes that result, and there may be antigens that are mixed and binding, creating these immune complexes that themselves can be injurious to the tissues. And lupus is an example where this is very prominent. And they also show that during chronic infectious diseases like subacute bacterial endocarditis, the immune complexes also can be very damaging. Now at bottom are a set of different diseases in which we think T cells are really dominant in pathogenesis, recognition of specific autoantigens, and inducing immune responses that result in injury to different tissues. And these are relatively common diseases like diabetes mellitus, rheumatoid arthritis, and multiple sclerosis. So we're going to focus a little bit more closely on multiple sclerosis, in which there are a number of different autoantigens that have been recognized, such as myelin basic protein and proteolipid proteins, and there are a couple of others that relate to the myelin sheath on neurons in the brain, and this can be a horrendous disease associated with brain degeneration, paralysis, and many other terrible outcomes. So to understand the immunologic implications of multiple sclerosis, we should just take a step back and appreciate that the strongest genetic associations for disease susceptibility, and this has been shown in genome-wide scans, are those that are involved in T cell activation. And in fact, the MHC class II variant DQ6 conveys a six-fold risk of developing this disease. So this all points towards T cell activation driving the disease. And mouse models such as experimental allergic encephalitis, EAE, has very comparable properties, and this again has very similar specificities of autoantigens that can be immunized and result in a disease very similar to multiple sclerosis. There's the targeting of myelin-associated antigens. And importantly, there is a prominent role of autoreactive T cells, and particularly Th17 cells have been recognized recently. And these have similarities to Th1 cells, but the production of IL-17 can stimulate the immune system in general and cause a lot of destruction. And recently there have been descriptions of prominent roles for IL-6 producing B cells, and in part this might be important because IL-6 is an activating factor stimulating a number of different cells, but it's also important for the genesis of pathogenic Th17 cells. So let's just look at the pathology of MS. And at left we see a very classical magnetic resonance imaging study using gadolinium enhancing dye, and we can see these white lesions that are shown in the middle, and these are T1 weighted GAD enhancing lesions in the white matter, very characteristic for demyelinating disorders. And here we can show a schematic or a simplification of what a neuron would look like with a healthy myelin coating at top, and then a nerve that has been affected by MS at right with damaged myelin. So this interferes with the transduction of signals throughout the brain and has important sequelae as a result. So what is the involvement of the adaptive immune system? And this is from a very recent review. And the concept here is that B cells may be recruited as they're newly generated from the bone marrow, and they may be selected for their antigen specificity in germinal centers and peripheral lymphoid tissue, as seen here. And as a result, the selected B cells and T cells, some of which may turn into memory B cells, and those go into plasma cells, some banked in the bone marrow, but others will enter the circulation. They'll be expanded further in peripheral lymphoid tissue, and then enter the central nervous system. They'll enter through vascular tissues, and there'll be a breach in the blood-brain barrier, and one can often see intraparenchymal perivascular infiltrates with these lymphocytic infiltrates, and there can also be what look like germinal center structures, which are not supposed to be in the brain. And why these are there and why they are expanded and what perpetuates them is not completely clear, but there are factors that are involved in physiologic immune responses, like B-cell activating factor, BAFF, also called B lymphocyte stimulator. And this is a B-cell survival factor made by activated cells, as well as CXCL13. This is a chemokine involved in attracting lymphocytes to sites of inflammation and sites in healthy tissue, just to be able to bring them into the right part of the germinal center. And all of these cells may be involved in communicating with one another. Some of them are released into the cerebrospinal fluid, and that has to do with, helps us make the diagnosis that there is infiltrating cells in the brain, and the classical finding of oligoclonal bands. These are immunoglobulin bands that show that there's a selective expansion of certain types of antibodies. And all of this reflects what is understood to be part of the immune pathogenesis of MS, and we believe now that this involves B-T cell interactions. There may be defects in regulatory loops, and there may be expansions of pathogenic lymphocytes, those producing IL-6 and IL-12, TNF, lymphotoxin beta, and other factors. So from the B cell's perspective, we can see here in the picture at bottom right that a B cell may have many potential contributing functions that are going on in multiple sclerosis. We've already discussed how there can be APC function, and there are B cells that can have specificity that will be expanded clones that are involved in recognizing these proteolipid specificities. And in turn, after correct or appropriate and maximal stimulation, there can be production, we can see it at lower left, production of lymphotoxin alpha and TNF that are involved in the formation of ectopic lymphoid tissue and potentially the formation of germinal centers that are involved in this autoimmune response. And B cells can make IL-4, which in some settings may even be protective against multiple sclerosis, or they can make IL-12, which can stimulate pro-inflammatory Th1 cells, or IL-10 that have other regulatory factors and properties. And at lower right, we can see that in fact a B cell might be making the right ratio of TGF-beta and IL-6 that will contribute to the selection and expansion of highly pathogenic Th17 cells. So all of these are theoretical functions that could be involved in the pathogenesis or the regulation of multiple sclerosis. What actual functions have been discovered, and importantly, what are the possible effects on T cells of B cell targeted therapy in multiple sclerosis patients? So examination of the tissues from multiple sclerosis patients have documented that there can be clonal expansions of B cells and plasma cells at different CNS sites. So something is causing antigenic selection and expansion. Therapeutic approaches have been tried. CTLA-4 Ig, which is a factor that has been approved for the treatment of rheumatoid arthritis, abatacept, is designed to block T-cell co-stimulation. And early trials with abatacept in multiple sclerosis were unsuccessful. In fact, it worsened the disease itself. So it just goes to show you that all mechanisms are not the same, and the pathways of diseases may be very different between, say, rheumatoid arthritis and multiple sclerosis. This does not seem to be a very attractive approach. In contrast, early trials with agents that target B cells through CD20, there have been trials with rituximab and ocrelizumab. These are very similar molecules, one a chimera, one a humanized, fully human antibody, which have been shown to deplete mature B cells through targeting of CD20 on the surface of these B lineage cells, but not really affect plasma cells, and it's been approved for the treatment of rheumatoid arthritis, has been tried in multiple sclerosis, and shown to improve clinical features and reduce GAD-enhancing MRI plaques in multiple sclerosis. And this has actually become one of the gold standards for therapeutic response. Importantly, it's been shown to reduce both B cells and T cells in the cerebrospinal fluid of treated MS patients. And in a very recent paper in the Journal of Experimental Medicine, they provided evidence in mouse models and in clinical surveys that they believe that, in fact, one of the major mechanisms by which this is providing benefits is through the depletion of IL-6 producing B cells in these treated MS patients. So they theorize that this results in a reduction of IL-17 producing T cells, and this is actually what reduces the pathogenic drive and provides clinical benefits. So just to review what we've discussed in this chapter, the adaptive immune system augments defenses with highly refined antigen receptors, and these are BCR in B cells and TCR in T cells. These enable distinct recognition of different antigen types by B cells and T cells, lymphocytes that themselves have very different functional capabilities. Together, antigen selection generates long-lived memory. So in health and in defense from infection, that's very important. However, if you generate pathogenic memory B cells and T cells, that may be very bad and contribute to a chronic disease that's very difficult to treat. In health, highly mutually reinforcing focused responses occur when B cells and T cells cooperate. And the adaptive immune system is responsible for susceptibility to autoimmune diseases when, in turn, it results in pathogenic focusing of inflammatory responses. In our last section, we reviewed how potentially we can find new therapeutic approaches by interfering with the specialized cooperation of B cells and T cells, and thereby this might provide therapeutic opportunities. I thank you for your attention and hope you've enjoyed the other sections of our program.